So in this figure, we have RNA splicing occurring. So in eukaryotic cells, RNA splicing is when a large portion of a newly synthesized RNA are removed. So what occurs in RNA splicing is that we have the removal of an intron, and we have the combination of exons coming together. So for RNA splicing to occur, we need RNA transcript, snRNPs, which is actually consisted of a protein and snRNA, which is short for small nuclear RNAs. And we also have the addition of other proteins. So when all of these come together, we have a spliceosome occurring. So what occurs in the spliceosome is that with the addition of more snRNPs, we have these snRNPs coming together and interacting with the intron itself. So when these interact with each other, the two ends of the intron come together and they join. And once they join, they are expelled out of the RNA transcript, and it's known as a cut-out intron. So with this removal of the intron, we have the combination of both the exon 1 and 2, which result in mRNA. And as well as snRNPs, they are removed as spliceosome components. I will be explaining enhancers and specific transcription factors. So what occurs here is that we have activators binding to distal control elements grouped as enhancers in the DNA. The reason they're known as distal control elements is because they are far away from a gene or they're located on the intron. In the second step, we have RNA binding protein, which brings the bound activators closer to the promoter. The bound activators cause mediator proteins to interact with proteins at the promoter. Nearby, we also have general transcription factors, RNA polymerase, and a group of mediator proteins. On the third and final step, we have these activators binding to a certain group of mediator proteins and to general transcription factors, which help activate the transcription initiation complex. These protein-to-protein interactions help facilitate the correct positioning of the initiation complex, and it will also help initiate RNA synthesis. Only when the complex is completely assembled can RNA polymerase move along the DNA template strand and producing a complementary RNA strand. In this demonstration, I will be explaining what occurs when TRP operon is on. TRP operon is essential for the production of tryptophan. When there's an absence in tryptophan, that means we have a repressor that's inactive. With our inactive repressor, this goes to bind to the operator, but it is unable to bind. The operator is the binding site of the repressor, while the promoter is the binding site of the RNA polymerase. When our inactive repressor is unable to bind to the operator, this results in allowing the RNA polymerase to continue on the genes of operon. These genes of operon then allow RNA transcription to occur. When RNA transcription occurs, we have the creation of polypeptide subunits, which make up enzymes. And these enzymes are then used for the tryptophan synthesis. Tryptophan synthesis then therefore results in the production of more tryptophan. When we have more tryptophan being produced, we then have the replacement of these tryptophan being absent. And this is all able to happen when our repressor is inactive, resulting in the operon turning on. In this demonstration, I will be talking about lac operon off, and I will also be mentioning repressible and inducible operons. So, what is a repressible operon? A repressible operon is one that is usually on. For example, TRP operon was a repressible operon. What this means is that the repressor would bind to the operator and it would cease transcription when RNA polymerase was no longer able to continue and it wasn't allowed to go on. And what is an inducible operon? Well, an inducible operon is the opposite, and it's one that's usually off. So what occurs in an inducible operon is that we have a molecule known as an inducer. This inducer binds to a active repressor, therefore making it inactive. This inactive repressor is not able to bind to the operator, allowing RNA polymerase to continue and make mRNAs. And an example of an inducer operon would be a lag operon. So in this situation of a lag operon off, what occurs is that our active repressor ends up binding to the operator. Once it binds to the operator, it seizes the RNA polymerase. It doesn't allow the RNA polymerase to continue on its journey. Therefore, the RNA polymerase ceases to keep on continuing, and no RNAs are made. In this presentation, I will be explaining what occurs with a lag operon on. So we begin with our lactose, which is also known as an inducer. This inducer or lactose binds to the once active repressor, changing its shape into an inactive repressor. This inactive repressor then does not have the ability to bind to the operator. Therefore, RNA polymerase has the ability to go through and scribe genes to mRNA. The enzymes for lactose utilization are then induced. LAC-Z codes for beta-lactosidase, which hydrolyzes lactose to glucose and galactose. LAC-Y codes for permase, which is a membrane protein that transports lactose into the cell. LAC-A codes for transaxicilase, whose functions are unclear. Adjacent to the lag operon, we have LAC-I, finally, which is the gene for the lag repressor.